The events required for a single round of cell division are called the cell cycle. When you have completed this exercise, you should understand the different phases of the cell cycle, understand the function of the S phase, understand the function of the M phase, and understand how DNA replication and cell division are tightly orchestrated to ensure that a parental cell divides into two healthy daughter cells. The process of cellular division to produce two daughter cells that are identical to the parental cell is called mitosis. Cells preparing to divide step through a number of coordinated activities collectively referred to as the cell cycle. The cell cycle has four distinct phases: G1, S, G2, and M. The two major events of the cell cycle are DNA replication and chromosome segregation. DNA replication occurs in the S, or synthesis phase of the cell cycle, and chromosome segregation occurs in the M, or mitosis phase. The S phase and the M phase are separated by two gap phases, G1 and G2. The gap phases allow the cell to check that it has the necessary materials and energy to proceed to the next phase. Checkpoints within the gap phases ensure that the DNA is undamaged before the S phase, and that the DNA is fully replicated before the M phase. If these requirements are not met, the cell cycle can be stopped. Before a cell can divide, it must replicate all of its DNA. This replication occurs during the S phase of the cell cycle. Two key events occur during S phase: replication and sister chromatid cohesion. During replication, the cellular DNA replication machinery duplicates each DNA molecule. Each resulting DNA molecule is called a chromatid, and the pair of duplicated DNA molecules are called sister chromatids. The sister chromatids are held together through a process called sister chromatid cohesion. The protein complex that links the sister chromatids together is called cohesin. Once the cell has duplicated its genome and has successfully passed the checkpoints of the G2 gap phase, the cell is ready to undergo mitosis. During mitosis, the sister chromatids are segregated to opposite poles of the cell. Each daughter cell thus receives an identical set of chromosomes when the parental cell divides. The process of mitosis occurs in a succession of steps: prophase, metaphase, anaphase, and telophase. Once mitosis is completed, the process of cytokinesis splits the cell into two daughter cells. When not undergoing mitotic division, the cell is in interphase. The cell is therefore in interphase during the G1, S, and G2 phases of the cell cycle. Note, although we have been showing the DNA as tight sticks for simplicity, the compact, condensed form of the chromosomes is only present in mitosis. In prophase, the long DNA strands that make up each chromosome wrap into a highly condensed structure in preparation for segregation. This is followed by nuclear envelope breakdown, which signals entry into metaphase. In metaphase, long fibers called microtubules emanate from microtubule organizing centers that are located at each pole of the cell. The two kinetochores at the centromere of each linked sister chromatid pair attach to opposite poles of the cell. This is called bivalent attachment, and it ensures that each daughter cell will receive exactly one pair of each chromosome. The microtubules then begin to pull the sister chromatids toward each pole. However, because the chromatids are still attached to one another by cohesin rings, the chromosomes align in the middle of the cell. In anaphase, the cohesion between the sister chromatids is lost, and the microtubules pull the two sets of chromosomes to the opposite poles of the cell. New nuclear membranes form around each of the sets of chromosomes in telophase, the final step of mitosis. Cell division is then completed by cytokinesis, the physical separation of the cytoplasm of the two new daughter cells. How well do you